JayAce is the guy you've been wanting to talk to, Taylor.
Yeah, I'm here. I don't know, if we got questions or if someone wants to bring up something.
If Taylor's not here, she asked that we DM her.
She's in the room.
Is anybody talking right now?
Waiting on you.
There she is.
Waiting on me. Okay, good. Is that I just heard everybody go silent, and then I was like, oh, I hope they're not addressing me.
Yeah, Taylor,
Is that what happened?
JayAce is here. He said he wants to talk to you about it now, right, Jace?
Yeah.
Yeah, absolutely. So I was doing a presentation on abiogenesis, and I was looking for some people to talk to and to critique some of my points. I wanted to get some feedback. And I was told about you because you apparently do like biochemistry research or chemistry research. And then I reached out to you in the DMs, and I'm just happy to see you here.
Okay. Yeah. Well, thanks. I do research in biochem, molecular biology. Usually pharmaceutical type of stuff, though. Actually, it's not what I'm doing right now, but that's usually what I do. But we could talk about abiogenesis.
Can you get us all discounts on? Can you get us all discounts on vaccines?
Yeah.
Part 30.
Hook you up.
You test on baby rabbits, John?
And Taylor, can I ask you a question quickly? Which side of biogenesis are you actually? Which side of the argument are you standing on? That we come from primordial soup?
No. So I stand on the side that undirected abiogenesis is untenable and unviable and undemonstrated.
Yeah.
So I personally study cell biology. So I'm not as keen to the chemical side of things and the chemistry. So I would like to get some feedback on the claims that I have made and the research that I've done on the chemistry in particular. I try not to focus too heavily on the chemistry side of it. However, it would be nice to talk to somebody who studies biochemistry and that could give me some critical feedback. Maybe one day we could do, I could ask Gia if we could do a stage or something and go through it. So from my understanding, you're from the position that abiogenesis is tenable and viable. Am I correct?
Yeah. Yeah, that would be my position. Yeah, I think, if you're looking for feedback and stuff, yeah, we could do something like that, like you said, a stage, just because right now I'm not necessarily in the right position to look at something and analyze it and give you feedback right away. But we could schedule something for that, and you can send the presentation or whatever to me beforehand so I could look it over, and then we could do a stage. And
So have you ever discussed abiogenesis with people before?
Yeah.
Have you debated?
In these kind of forums, personally, I don't do origin of life research. Like I said, my main research is in the pharmaceutical industry, and right now I'm doing some stuff with fossils, but it's a little side project. But I've talked, not formal debates, but I've talked with people in these kind of forums, and just I haven't debated, but I've spoken with people at my university and whatnot who actually do origin of life research and stuff. So that's my experience with it.
Dude, that's really cool. Do you know any origin of life researchers that'd be willing to talk to me?
Do you know
JayAce always tries to school us on that. What do you mean, JayAce? I thought you were an expert on this.
Yeah, dude, I've talked to you about myself.
I have an understanding of it, but it's not my active area of research. Like I said.
Also, now you're scared because it's actually some.
He can feel the trap.
The last thing I saw on your.
I never claimed that I was ever doing active research in the field of origin of life. I've always just done these debates talking about the research that I've read from those fields.
Yeah, but you know all about this. You're like
discussing
well
it.
informed
It's
on
okay.
this, right?
It's okay. It's okay. It's like it doesn't matter.
Now, hold on, Taylor. We have a boat to pick because he's been schooling us that they've made
yes.
Okay, so
I don't have to
cells
be
and
an active. I don't have to be an active origin of life researcher to give you guys the experience and knowledge that I have with it. Like I could still do that even without that being my active area of research because it has a lot of overlap with biochemistry and molecular biology.
it's
hey,
okay.
left
It's
lane. Your
okay.
boy sounds scared. Left lane.
Okay, look, it doesn't matter. Like I like a fresh start with anybody. So what everybody says, it's okay. I'm just talking to you like as if it's our first time talking. I don't know nothing. So what I was just gonna ask you, Jace, is if you would be willing to, you know, debate or discuss abiogenesis with me. Like on a live stage.
Yes. We can do that. Yeah, like you're talking about here, like on a Discord stage thing.
Yeah, that'd be good. But if you don't feel comfortable debating it, I wouldn't want to like pressure you to do it. You know, if it's something that you actively debate, then I would feel comfortable because I wouldn't want to like you know, I wouldn't want to like come at you knowing more than you do and feel bad. Like I wouldn't want to do that. So that's why I'm asking if that would be something you'd be comfortable debating. But if
Yeah,
not,
I wouldn't mind debating it. That would be fine. Like if you want, like I said, though, if you want to have like a kind of like a formal, not like a formal debate, but a debate, like that would be something you'd have to like, you know, give me a time and date so I could kind of plan for. But I'd be down to do it.
okay, only if you would be comfortable doing it. Like I said, like if that is something you'd be comfortable doing, we could absolutely set up time.
Yeah, certainly.
Okay.
Just DM me.
When's
and
the
we'll
next
try to
time
work something out.
Are you available tomorrow night or tonight or like when's your next availability?
Today, Monday. I would say probably Friday night, Saturday afternoonish.
Okay, Saturday afternoon, like next Saturday.
Yeah.
Okay, yeah. Well, I can ask you about that. And so if you don't mind me asking, what is your hypothesis on origin of life?
Well, I wouldn't say that I have like a single hypothesis, right? Like the current thinking is like an overlap of a bunch of scientific fields with like individual steps and then overall steps and mechanisms, like linking them together. So I'd be happy to discuss those. I think it's too complex to boil down to just a singular hypothesis. I think there's many hypotheses that describe the process, and we can discuss those.
Okay, yeah. So I was just kind of asking like protein first, RNA first, protein RNA first, metabolism first. Is there one that you like hold to more tightly than
I
others?
fall more on the RNA.
Okay, RNA.
Not
Yeah.
necessarily the RNA world, like the strict RNA world hypothesis, but I lean more towards RNA being the first like macromolecular biomolecule that emerged.
Okay, so like the ribozyme theory.
Yeah, similar to ribozymes.
Okay, yeah. Well, I'll talk to Gio and maybe we can talk next Saturday.
Yeah,
Or this
sounds
upcoming
good.
Saturday. Okay, sounds good.
Tell him, or I think he's here. So like in between like 1 and 4 p.m. Eastern time, if you don't mind, for like a start time.
Okay, yeah, that'd be good. Maybe like 2 p.m. Eastern. Would that work?
Set it up.
That could probably work.
Okay, yeah. Okay, I'll look at my schedule. I think that would work. And I'll DM you. Just make sure you send me a friend request so you can get those DMs.
I'll bring the beer, guy.
All right,
I'm bringing the
sounds good.
popcorn.
That still has a T-shirt with JayAce's face on it. Jace, you seriously like try to downplay yourself, like, oh, that's not. You know what I'm saying? You know all about this stuff,
Just
bro. Don't
being
front.
honest, right? I don't want people to start saying that I claim I'm an expert in everything. I have a vast deal of knowledge about abiogenesis. Don't get that wrong, but it's not my active area of research in my professional.
Well, you better study up,
sense.
Jace, because I'm bringing James Tor to the debate. He's a personal friend of mine. He's gonna hit you upside the neck.
Oh,
with,
please bring him.
I'll bring Professor Dave.
He says he wants all the smoke, bro.
Hey Taylor, so what do you think about the debate between Professor Dave and James Tour? Have you seen that?
Taylor was trying to get it in tomorrow. She said, "Let's get it popping tomorrow." What's up?
Well, I would actually like to debate him tonight, but you know,
She said,
it's
"It's
up to
on
you.
site."
She said
It's
right
on
now.
site.
Shit's
It's
going
on
down right
site.
now, Jace.
Taylor, can you answer my question? Like, what do you think
Yeah.
about?
Well, we can discuss it. I mean, I don't mind this. I'm
You don't want
out
that.
walking right now. Right, I
You're
can
always walking the dog.
discuss things.
I think that
Okay, so
dog
I
wants
don't
you.
have the dog, man. I brought the dog back inside.
okay, so my question is, what's your opinion between debate of Professor Dave and Dr. James Tour?
Yeah, I thought it was an interesting debate. I think that it'd be better if the crowd knew what they were talking about because there was like very specific claims brought up that like Dr. James Tour was like asking him to describe like the chemical mechanism of how it would happen. And then Professor Dave was just like pointing back to the research article title and it never really got into like the specific chemistry of it. So it, you know, it like we never really like hit like a high point in my opinion. Like it was just kind of like these like different topics that were brought up that we never actually got into the weeds into. So I don't know. I felt like that part was kind of lame because I would like to see Professor Dave actually talk about specific chemistry. But yeah, it was interesting.
Yeah,
So, so do
he's
you
not a
agree like
professor.
what James Tour was saying continuously, like go and draw, go and write it on the blackboard or something, and he was saying, so who lost the debate according to you?
I mean, I think that Professor Dave is better at like the talking part of the debate, but Dr. James Tour is definitely more like knowledgeable of the chemistry. And that was apparent. So yeah, I think it was funny how Professor Dave wouldn't go like get into specific chemistry with him. So yeah.
But since you guys are mentioning Professor Dave, there's one because I'm like 60-40 leaning globe, and I'm not really sure where I'm standing. But since you mentioned Professor Dave, there's one specific argument that I do not know the flat Earth alternative of, and that is the different spinning of directionality of the constellations on the different spheres, right? So if you're standing on the South Pole, you would see the stars and whatever rotating one way, and on the North Pole, it would rotate counterclockwise, as opposed to the rotation you would see on the South Pole. And this wouldn't make any sense on a flat Earth. If you're looking at a specific part of the sky, it would all appear to rotate the same way, right?
Well, this is the hard transition. This is a hard transition. Are we done with the? Or Taylor, do you guys want to do a little sneak preview now, or we can get onto your thing? Hold on. We can get onto your thing about the different rotations and stuff, but let's just like try to conclude this as much as possible.
Yeah, I'm sorry. I just jumped in. I do not know where you guys were having
No,
the
you're good.
conversation. I'm sorry.
You're good. Just hold. Yeah, no, we get to it right after if you bring it up after this.
Yeah, I would like I said, I'd be willing to talk to Jace whenever. Like, I would be willing to talk right now. It doesn't seem like he's in a position to do that. So it's hard to kind of get into the weeds of something that we can't like finish, you know, talking about. So just as soon as you can, if this upcoming Saturday is as soon as Jace, they can do it, that's fine. But like, it's really up to him, Jeremy, you know, like I'd be willing
He
to
can
talk
do it
about.
right now. He's already in the weeds walking around. Go ahead, Jace.
You funny dice. So, well, right. It depends on what you want to talk about specifically, how deep you want to get. Like, if you're talking about going over the chemistry, like
4
James
hours.
Tor wants to do, like we can absolutely do that, right? But I need a little bit of time to prepare. I have some articles, right, with the detailed chemical mechanisms, and we could go through the chemical steps, but that's going to take some preparation. If you want to just talk about like the overall, you know, like certain events being witnessed, like RNA being naturally encapsulated by phospholipid mono and bilayers, like the overall steps we can just discuss. But if you want to talk about like the minutia of the chemical specifics, right, that's the thing that's going to take preparation.
I think I think you should get a Bible ready, Jace, in case you change your worldview at the end of the conversation. That way you're kind of, more prepared for the future.
Well, just so you know, on Saturday, when you do end up having this debate, it's 6 hours for the debate, and then we do like a 7-hour Q&A. So clear 13, 14 hours out to be safe.
Can I perform at halftime?
We'll do a couple. Let's shoot for a couple hours, not 16 hours, Jeremy.
That's what they shot for a couple hours last time. Yeah, we'll see. Yeah, no, we'll see how it goes.
Okay, so I'll just ask you, I guess, about one of the claims you just made about the phospholipids being encapsulated by the RNA. So, my question would be, first of all, like, how did they form these phospholipids? You know, and then I would ask you about, once it did encapsulate RNA, how would things move in and outside of these phospholipid bilayers without transport membrane proteins?
So it was a group. There's been a group in America and a group in Australia. I posted in this chat before some of the papers. I'll have to look through because I forget the name. Dale something, I believe, was the first name of the guy. But nonetheless, what they did for these observations was they went through testing these different like prebiotic ocean and just regular water conditions. Like they altered the conditions. Hang on a second. I'm so sorry.
You just leave the dog poop, Jace. Don't worry about picking it up, bro. Just keep it
I'm
moving.
not interjecting while he's Googling. Aren't my brain force a thing, like protein channels or whatever?
Yeah, this is way too complex for you to just Google in a few seconds and think you're going to be able to summarize it.
Obviously.
But nonetheless, they go through making the water mimic these prebiotic hypothesized conditions, right? And then it's just the formation of phospholipid mono and bilayers is just thermodynamics. When you put those chemicals into water, they form these vesicles, or they can even form like micelles, which is usually used to describe like soap, but they do this automatically just through thermodynamics and entropic purposes. So as they do this, they will encapsulate certain chemicals that are in the water. And so if there happens to be RNA strands or whatever else in the water, they will get encapsulated during the process. Though some chemicals can still pass through, they're called semi-permeable membranes, which allow certain chemicals to pass through the membrane, going from the inside of the micelle or the vesicle or the bilayer, monolayer, whatever it happens to be, to the outside freely. So there's multiple ways that it can happen, and it's been observed by multiple groups now. But the first two was a group in America that I believe they did it in freshwater conditions, and a group in Australia did it in saltwater conditions and observed RNA strands being encapsulated.
Yeah, I acknowledge that. I acknowledge that process could happen. But the only thing that I would question is how viable would this membrane become? How viable would this membrane be in terms of being able to transport things in and out of it and undergo some sort of metabolism where like you could have like electron transport throughout the membrane without it breaking down. So that would be one of the main things that I would be wondering. And would it allow in hydrophobic or hydrophilic compounds or both?
Well,
Like,
we're
you know what I mean? So
talking, you're already going, you're like way too far ahead in the process. Or we're talking about the formation of how the first like protocells could have formed, and all a protocell is defined as is a macromolecular biomolecule encapsulated in a lipid membrane. Doesn't even have to be a phospholipid. It's any kind of lipid membrane, right? And so we're talking about the witnessing of the first step of like protocells actually forming, right? From there, that's when you start to get they actually, the group in Australia, I believe, started to observe processes with these RNA encapsulated vesicles that were similar to biological evolution, where as they replicated themselves, these RNA molecules, the ones that were better able to put up with the stresses of the environment were the ones that would replicate to the next generation, and the ones that couldn't, like say there was some kind of acid or some kind of base in the water, and that RNA strand didn't have a sequence that could neutralize that acid or base, it would be destroyed. So they were already undergoing these kind of evolutionary processes straight from the bat, and so that's how these further structures would develop, and there's a lot of steps involved in that.
Okay, I see what you're saying. So, is it that you define a protocell as just like a biomolecule encapsulated by a lipid membrane?
Yeah. So that's
I'm
like
just maybe
the
you could
first
share with me
step.
where that definition comes from, because I would imagine that a protocell would need some type of reproduction mechanisms.
Yeah, they do. RNA strands are, like you said, they're able to turn into something akin to, when we're talking about these early ones, they aren't actually ribozymes, but they're molecules akin to that that can self-replicate. We're already past the autocatalysis point. Like the RNA molecules are already self-replicating.
Okay, that would be really cool if we could actually get like a self-replicating RNA from scratch, to assume that.
Well, it's not an assumption. They have done it. And not just in these kind of experiments, these autocatalytic cycles, there's a whole bunch of them that have been discovered just in pure chemistry. But when it pertains to biochemistry, there's multiple different parts of this abiogenic process where these autocatalytic or self-replicating RNA strands have been observed coming about naturally. It's not something that has to be assumed.
Okay, so do you know the materials and methods in which that was able to happen? Just a general, not like all of them. I'm not asking you to go really in depth with it, but just like a general, like what they did in order to get this self-replicating RNA.
Yes. So I mean, it's a chemical process, right? It goes from inorganic precursors. So they sterilize the water, basically. They get rid of all current life forms that are in the water in an region that would suffice for the experiment that they're going to do. So that's step one. Then they recreate these different prebiotic conditions. So they add certain chemicals like methane or sulfur dioxide, or just to try to test these different prebiotic condition hypotheses. And then they add inorganic precursor molecules, different ones that would have been hypothesized to be present, things like H2S, um, methane carbon dioxide, hydrogen sulfide, um, carbonic acid, these different chemicals. They go through ranges of them, but you have inorganic chemicals that leads to organic chemicals. So those are carbon-based organic molecules. Those lead to the biomolecules, eventually the monomers, the nucleic acid monomers, or the RNA strands, and then the actual RNA strand is the polymer, the actual monomers linked together, and it comes about through like these different chemical steps, and there's a lot of minutia that's involved in all this stuff, but that's how they get to the RNA strands, through chemistry, basically.
Okay, so you're saying they started with inorganic chemistry and made self-replicating RNA?
Yes.
And that's not true.
It
That's not what
is.
happened.
I've
Okay, well
posted
then,
the articles in this
okay,
chat before.
give me a title to what article you're referencing at this moment.
Give me a second. I'll see if I can pull it up.
Even the author or whatever.
So what's your take, Taylor, that the RNA, in order for it to be self-replicating, has to be like sequenced in such a way unnaturally?
Well, for example, they didn't just start with inorganic materials. They have separate they have separate experiments where they're making activated nucleotides from inorganic materials, and then separate experiments where they're polymerizing nucleotides, and then separate experiments where they're actually using templates and activated nucleotides in order to make these RNA molecules that can perform this behavior. And then on a separate experiment with the self-replicating ribozyme, they used an already existing RNA polymerase, tweaked it up, and got it to have ribozymic activity. But I think that what he's referencing is kind of like a melding of these experiments.
Yes. This group in Australia, the guy's name is Dale. I'm looking for the articles that I posted. I'll have his last name. But yeah, they did kind of a mending. But yes, they have done it in a step-by-step way, right? Because you want to elucidate the mechanisms. So they've broken down like going from every molecule, like these different pathways of how these inorganic compounds could have formed these RNA strands. Some parts of the process involve like catalysts, like certain pieces of clay that are hypothesized to have been present on the prebiotic earth, like montmorillonite clay. So yes, there has been like where they've done it individual steps, and then this group in Australia had put it together in, I believe, a saltwater environment. But I'll find the articles and refresh on that.
Okay, so is it called the process of RNA synthesis catalyzed by montmorillonite clay?
Yeah, but that's a study just breaking down one individual step, right? So the idea there is if you can work out every individual step, maybe you can't by yourself or in your lab or out in the field with just your team create the whole process, because it's a whole bunch of steps that require different kinds of conditions. But if you can recreate each individual step, it's fair to say that you've worked out the mechanism for how these steps link together, right? But there are groups who have done these like precursor molecules to biomolecules. So those are out there too. Like, I don't know why you're saying that, but
no, I did acknowledge that. I acknowledge that there's different experiments doing different steps. However, the question I asked you was, is there an experiment where they use inorganic molecules to form self-replicating RNA? And you said yes. So I just wonder where that.
Yeah, well, this group did they published a few papers, and each individual paper describes certain parts of the process, but they went from a sterile aquatic condition to biomolecules, right? And they wrote up how they did that, and it was going from precursors to those RNA biomolecules that got encapsulated and started undergoing these evolutionary processes, right? They did a few write-ups describing all that.
Okay, so yeah, the question I was trying to ask you was if there was one experiment that went from inorganic molecules to self-replicating RNA. And you said yes, but would you acknowledge that was actually separate experiments?
Yeah, they're separate experiments. So
Okay.
I didn't mean to put it in that way. Though this one group kind of carried out all these separate experiments, all kind of in one scenario.
Have you looked at the conditions and the precursors in each of those experiments? And if you have, like, have you seen that there's like differences in the environmental conditions for each step?
Well, they try to make the conditions. There's different, like, geologists have different hypotheses of the prebiotic conditions. So they try to make the water, they'll have different sets of conditions. They'll do the experiment with the water being alkaline, with the water being acidic, with it being reducing, with it being oxidizing. Right? They'll change the environmental conditions, and then they'll also change like the precursor molecules, like if they put sulfur dioxide in there or carbon dioxide. water vapor, things like that. They'll change those molecules, ammonia, hydrogen, like what the precursor molecules are. So they go through an array of these different environmental conditions and like precursor conditions.
Yeah, it's kind of weird, like that they have to have different conditions for each particular step. Like, why wouldn't they just try to keep the conditions the same for each step? If
I
they're
don't think they
assuming this
change
is
it for
the
each. I don't think they change it for each step, but a lot of these polymerization reactions, like to form, whether you're linking amino acids together to form proteins biotically, or you're linking nucleic acids together to form RNA or DNA, they require changing conditions. It doesn't occur under just like one stable atmosphere and stable condition. Like you have wet and dry cycles, you have different chemicals coming in and out of the environment. So they try to mimic that in these experiments. They try to, there's different hypotheses of what those chemicals and what those conditions were. So they'll run different experiments under the different hypothesized conditions.
Okay, so yeah, it's funny how like the wet dry cycles, that would just be like they did that just for like the formation of proteins. So it's not even like that they would need wet for one and dry for other. They actually need wet and dry for like forming one particular biomolecule. So even within the formation of one biomolecule, you'd still need different conditions. So and then imagine adding other molecules. So they're having to put so much pressure on the environment to form even the most basic biomolecules. And then also another
Well,
question is how would
yeah,
they have
you have
purified?
to.
So like remember you mentioned how they have like this purified water because like if you have RNA or DNA in like dirty water per se, like it'll totally like break down. Like that's why you need to have like hyper pure deionized water. So like how would we have this hyper pure deionized water on early Earth?
Well, no, the reason why you sterilize the water is because if you don't, when you put things like hydrogen sulfide, hydrogen gas, sulfur, nitrogen, ammonia, any of those precursor molecules into the water, if it's not sterilized, the life in that water is going to use those molecules. It's going to absorb those molecules and use them. So they won't be any good for you in your experiment. So you have to eliminate the life forms that are in the water
Yeah,
currently
but you could just
so that they
autoclave
don't
it.
disrupt the
Couldn't you just
prebiotic
autoclave it to make sure there's
conditions.
no life in it?
No, there's certain chemicals. There's chemical cycles that you go through to eliminate certain things. Like if you're trying to get rid of metals versus if you're trying to get rid of bacteria versus if you're trying to get rid of fungi, right? There's different chemicals and different processes that they'll cycle through. If you're trying to get rid of bacteria, life, other RNA, DNA, strands that could be present in the water, that you use something called an RNase or a DNase, which are
I'm familiar.
enzymes that will chop those up. Yeah.
Yeah, I get that. I get that. And so you wouldn't want to have any contamination because then in your results, whenever you're analyzing for RNA, you want to say, hey, we've identified RNA here, and it's not because there was pre-existing life forms in the solution. So I totally understand what you're saying. However, have you seen that whenever like you try to store DNA and RNA, you need it in like purified water? Are you aware of that?
Yeah, but it can be stabilized. Like I said, certain clays will stabilize the RNA, which RNA is more stable than DNA to begin with. But there's certain conditions that will stabilize the RNA or the DNA strand, right? When you have it in like your chemical lab, yeah, it's not going to last that long because the conditions aren't. They're not attuned for it lasting that long, but you can crystallize it and it'll last much longer in the solid state. So that there's ways to enhance the stability, right? And
But
nature
they have
does
to
it
have it.
on its own.
But this has to happen without any enhancement by or intervention by humans or any
It
sort of
doesn't.
intervention.
It doesn't
You can
because, like
rock
I
it up with
said,
baking soda too, right,
the
Jace?
the Montmorillonite clay thing has been observed countless times now.
No, I'm familiar. I'm familiar with that. I'm just saying,
catalyzing and stabilizing RNA strands.
is that the paper that you're referencing in particular? Like, is that the one that you're?
No. That's all
Okay,
entirely.
get.
The people who did the Merlinite thing are an entirely different research group than the ones who did the RNA investigation thing.
Yes, I'm aware of that. However, like for the matrimorionite clay, they actually like utilize like activated nucleotides in that. So I mean, is that really prebiotically plausible?
Well, there's more than just one group that's done this. Like maybe the original group that could be true, but there's certainly other groups. There's people at my university that are doing these mineral catalysis, working out these cycles with these minerals.
Is there any way that they would talk to me about that? Would they be willing to ever talk about it?
Yeah, I could ask them.
That'd be really cool. Okay, back to the clay experiment. So, like, I think that it's important that we like target individual papers so we can understand, like, because if you're talking about one paper and I'm responding about the materials and methods to another paper, then we're just kind of running in circles. So, like, for the clay one,
Yes.
for the clay one in particular, one of the interesting things that they found, this one was called matrimorionite catalyzed RNA synthesis. And so they had a couple interesting things in this paper. One of them being that like they used activated nucleotides. Also, they found that in aqueous environments, the nucleotides and RNA oligomers.
Hold on. So, sorry to stop you. So they used activated nucleotides. Like I guess that's your big hiccup with these experiments. Then they may have all done this because all that means is that they used nucleotides that had the ability to have chemical reactions, right? And that happens naturally, right? There's certain elements that are just in nature. They're in water. They're in the environment. That activate nucleotides if they're present. They make it so that the nucleotide can react with less activation energy.
Mm-hmm. Yeah,
So
then
the
they
fact
should
that they
demonstrate that.
use activated nucleotides isn't like some like hiccup in the experiment, right? It's just it's literally just chemical lingo.
But didn't they acknowledge that as a limitation?
I don't know why they would. Why would they acknowledge that as a limitation, right? If you're not using activated nucleotides, then you would expect nothing to happen.
Right. So
Yes.
this is the
So
thing,
that
like
doesn't make sense.
if it's plausible to have activated nucleotides on a prebiotic earth and then to get them to form, then that should be easy to demonstrate.
Yes. And like I said, this has been demonstrated in a step-by-step basis because it requires different conditions for each of the steps. You're literally going through like the different kinds of chemistry, going from inorganic to organic
Yeah,
to
but
biochemistry. So
you don't think that's
like
just another
it's different
impossibility though, Jay?
environments and different processes that have to arise, and not everybody can replicate all that in one experiment.
Doesn't that just seem like another improbability though? Like another issue? Like if nature
No,
has to. Okay.
it's just understanding emergence and that nature is complicated, right?
Well, I'm just saying, like, in order to have the necessary conditions for these prebiotic molecules to be established into macro bio molecules, and you're saying, and she's addressing the fact that each stage of the condition changes. That just seems like it's just making a lot more complicated for nature to arbor these sorts of conditions required to have the outcome be suitable for life to be had.
I don't know, the fact, like I've done so much chemistry. I guess I'm just acquainted that certain chemical reactions require like a certain set or a certain range of conditions, and sometimes those conditions are found in certain areas of nature. Like that doesn't surprise me, that going from these simple inorganic compounds, basically elements, to a complex protein or nucleic acid, that would require different conditions and different processes doesn't surprise me.
Yeah, but I'm just saying. Oh, go ahead, Taylor.
But it doesn't surprise me either. But then to make a cohesive story as to how life could have formed, it's a little bit surprising to put all these different environmental conditions for different parts of the cell on the back of nature, who's not, who doesn't have an end in mind, So that's one of the things is that, if this was plausible, they should at least be able to replicate it in some prebiotic plausible conditions that can move from point A to point B, and at least have like some sort of cohesive storyline as to what could have happened.
There is, right? So I think that the best thing to do, I'll post some of those papers. I'll look for them when I'm done talking, because Discord changed the way it is on a phone, so it's like hard to talk and search the text. So I will afterwards, but you should send me, for our debate on Saturday, send me a few papers, like 2 or 3, that you want to talk about, right? And I'll read them, and then we can actually talk about the specifics. That would probably be the best way to go.
So that's the thing, like, I don't have like a specific storyline that I'm like, because I would need to know what you think happened. And then I can send you the papers that would best suit your position, and then we can talk about them. Because I want to give you, in terms of papers, the benefit of the doubt. So I would need to know what is your optimal storyline, and then we could go from there. However, I don't think that there is an optimal storyline, so I wouldn't have like specific papers or original life research to reference, because it just depends on what you think happened, because there's competing theories. You know what I mean? So if you're saying it's plausible, I would need to know, okay, well, how is it plausible? And then we can talk about like what papers show the best optimal case for your position, and then we could talk about those papers. So yeah, if you want to say like RNA first, I can try to send you some of the papers on the nucleotides. Yeah.
Yeah. Right. The thing is, it doesn't really matter, right? Like what I think. I gave you kind of an overall kind of thing. The general origin of life research thinking is, right, it's this intersection of a broad range of different sciences like geology, physics, chemistry, biology, meteorology, climatology, etc. All these things, where they meet, that's where this origin of life research lies, right? And the overall thinking, right, because I'm a biochemist slash molecular biologist, I tend to focus on the like protocell level, the chemistry that led up to the first thing that we define as protocells. And that is inorganic compounds performing chemical reactions, leading to organic compounds, leading to biomolecules or biocompounds.
Can I ask you a question, Jay?
Right. That's the overall thinking in the origin of life field, based off the experiments that have been performed on those steps. It's not really the thinking, that's the conclusion of what's happened according to the people who have performed those experiments. They don't have like every step worked out exactly, but they have options of how certain steps, have mechanisms of how these steps could have happened, right? And
Can I
they
ask
all
you a
make
question, Jay?
sense to me. There's no discrepancy in any of these possibilities. So though I can't give you a specific of how every step of the way went, the overall picture seems pretty clear to me.
Okay, yeah, just share with me whatever picture you think is clear, so we can discuss it. But yeah, go
I
ahead, Dave.
already did. Just did.
All right, go
Jay,
ahead,
can I
Dave.
ask you a question? Oh, someone else going? Go ahead.
No, I said go ahead.
You go ahead.
Yeah. So what's the shelf life of these inorganic compounds in order to get to these steps down the road where we have the necessary compounds for life? So what's the shelf life?
The shelf life of
What do you
inorganic
mean?
compounds is going to be much higher than organic compounds. I just want to throw that out there.
So for like the formaldehyde
Purified inorganic
and
compounds, generally they're stable,
Yeah,
but
they're
I don't know about
inorganic,
that.
you have to know the difference.
traces of biogenic carbon are found in fossils and rocks, not fossils, rocks that date back to like just after the Hadean. So like inorganic molecules would not have come anywhere close to breaking down come that point
So
yet.
I think what he's trying to ask is what's the shelf life of like organic compounds like RNA
Well, no,
or
I'm asking like the formaldehydes and the amino acids, like the base compounds in order to have the base parts for life to emerge. So
Mhm.
yeah, like these are some of the necessary requirements, and they have shelf life. So I'm just trying to get an
so amino
understanding on
acid
how
would
long.
It's the polymers. The polymers are the only ones, the proteins, the DNA, RNA, the carbohydrates, the phospholipids, the overall polymers are the only things you have to worry about with these half-lives of them breaking down. And like I had discussed before, all the precursors to these, the nucleic acid monomers and the amino acids and those things, they're half-lives and stuff are way past the time frames that we're talking about here. So
Okay,
the really
like
it's
years or
just
something.
RNA and DNA that people for some reason there's this misunderstanding that they have that they're unstable, and I think the pharmaceutical companies kind of propagated that with the mRNA vaccines or whatever, because those do have to be stabilized. But like I said, with these abiogenesis origin of life experiments, there's been minerals, clay out in nature that has been shown to stabilize RNA and DNA strands. So they're just natural processes.
that would need like a low water environment.
Well, for like the amino acids themselves, though, are you saying they
What do you
don't
mean?
degrade?
Amino
Or
acids have a huge half-life, die slow. They're not
I'm
going
I'm
to
talking in nature, not
degrade.
in your lab, though. Yeah, I'm talking in nature, right? Exposed to the elements, right? You're
Yes,
saying that
they're
they
still
don't.
not going to degrade, man. Not in a prebiotic condition.
It's the polymers like the proteins, the RNA, the DNA, that would have like a much shorter half-life than the monomers, which would be like the amino acids. Nucleotides can be iffy too that make up RNA, but the
I don't disagree with that. Yeah, I don't disagree
yeah,
with
yeah.
that. My gripe is within the base ingredients for life to be had. Like all of that stuff has to be in the right sequence, in the right conditions, and then there's a shelf life for those things. Like if you're just gonna say that they don't degrade, I mean, I don't know what to say to that, but I would think everything degrades
They
over time,
they do
including these things.
degrade. They do degrade, but their half-life is on like it's on astronomical scales, man. The like I had said and like Taylor said, the only ones you'd have to worry about degrading due to their half-life is the RNA or DNA.
All right, well, I'll just leave it there for now.
Yeah, that's a good question though. In terms of
But even that, though, Taylor, even that, right? I have a machine in my lab that basically can make DNA or RNA strands for me. I just put the chemicals in and put the
it's
sequence
my
in that I
PCR.
want.
PCR.
No. That's an amplification. This actually
Okay,
does it. It basically does the chemistry for me
you're
to
making
actually.
plasmids.
Well, it can be used for different things, but I'm just talking about making the DNA or RNA strands themselves, like linking the nucleic acid monomers together into the polymer. We have an instrument that does that for us. So because
Without
it
polymerase?
would be lengthy if we didn't have, like, you could do it on your own, but it would take quite a bit of chemistry and time to do. So somebody invented this instrument that does it for you. It makes, it'll make 50 million DNA strands overnight for you. That would take you a month as a grad student to
Without
do.
polymerase?
No, it uses polymerase.
Okay, so use the enzymes.
It's not a prebiotic machine. This is just for chemical purposes. It's just to crank out as many DNA, RNA strands as you can, fast as you can, without having to do all the chemistry yourself.
Yep.
But
so wait,
so we have a
have you?
machine that will do that, and based off of the conditions that I store the DNA in, never mind having it out in nature interacting with minerals and clays and all sorts of different things, but even if I just change the conditions and that I'm storing it in my lab, make it more acidic, less acidic, more basic, less basic, more oxidizing, less oxidizing, etc., that will
That's what I was saying.
increase or decrease the stability, the length that the DNA RNA is going to last. And you can increase it if you do the egg drop method to crystallize it. You can increase the half-life quite substantially just in a lab condition.
And that
I
doesn't
agree.
happen with the amino acids and the formaldehydes. Like if you change the temperature or the pressure or the whatever exposure is, it's to, because it's like we're dealing with nature, right? Like we don't have some sort of a sterile laboratory environment where things are being separated and mixed at the proper times and such. So
Yeah,
I'm trying to
exactly.
wrap my head. Yeah, just trying to wrap my head
Well,
around that. Like,
that chaos is part of the process, Di. So all these different
right.
changing conditions in nature, right? Because
No, I
the
understand.
different parts of the chemical process require those different conditions. Like step one may require the presence of an acid to be there, an acid catalyst. So you need something like CO2 dissolved in the water to form carbonic acid to catalyze the first step of the reaction. But the third step might require like a base. So now nature needs different conditions to come in, right? So and that's just all part of the chaos of nature.
Right. Well, that's kind of what I was touching on, right? Like the same thing that you need, right? The dynamic system to kind of keep all these different things happening for the right sequence to happen at the right time to create the next building block, so on and so forth. Is seems to be like the same thing that could work against you in the sense that you called it chaos, right? There is no organizer, if you will, or conductor, if you will. It's happening on its own, which just, in my opinion, leads to the more improbability of it
Well,
happening at the right.
it's all entropy driven, right? The more complex a chemical system is, the quicker it can dissipate an energy gradient. So when you get a system like the prebiotic earth that it is getting the sun's rays, the sun is imparting energy on the earth, right? The more complex the chemistry gets on the earth, the better, the more efficient and faster the entropy can increase into the environment, basically. It's all
And that would be the more faster that
driven by
certain
entropy.
molecules can break down.
Like RNA and DNA, proteins, carbohydrates, glucose, those kind of chemicals they dissipate entropy much more efficiently and faster than like ammonia or oxygen or nitrogen or sulfur would do.
What
Have we
what
tried
what was
these
the
like
term?
static environments though in original life research?
Yes. They've
And
gone
they and
through
they and they stay stable?
there's been tons of experiments done and tons of different hypothesized prebiotic conditions pertaining to those experiments. They don't just go with like one, like, okay, the Earth was thought to be this one condition. No, there's different hypotheses, and when you're doing origin of life research, you test all the hypothesized conditions that are pertinent to your experiment.
What was that term you used? It was like a macromolecular. Yeah. Yeah.
Sorry, Amazon
Something that
just
would
arrived
seem.
at my door.
All right, tell him I said hi. Something that would seem like oxymoronic to say, right? Like
Yeah.
macromolecular. But
Well,
the thing is,
it's
like
in the
these sporadic.
context of other chemicals, right? It's describing its size in the context of other chemicals.
But like, so you're saying like the sporadic environment would be needed to, form these different polymers and things of that nature. However, the sporadic environment would be the very thing that breaks them down at the same time.
Well, no, because we know that, like I said, these things have been experimented upon. We know under what conditions a lot of these steps occur in, and it's not that like just because the conditions change everything falls apart. No, the change in conditions is what leads to the next step, the further building of the chemical system.
So right, so like the wet and dry cycles, for example, are needed to like form different polymers in origin of life research. However, you would need it to consistently be dry to keep the RNA stable in this morelinite clay in their
No,
experiment.
no, because you can
Yeah,
have
because
like an acidic aqueous environment that might stabilize certain RNAs, or you can have an
but you wouldn't
aqueous.
have an acidic environment for things like amino acid formation. For their amino acid formation experiments, they need a highly like basic and reducing environment.
Yes, yes.
Okay, so that's an incompatibility.
No, because
No?
the amino acids don't just fall apart just because the conditions change. They need certain
Not the
conditions
amino
for their
acids,
formation.
the
What
proteins.
the proteins don't fall apart either.
So which
Just because
okay,
the
so
conditions change.
in the experiments where they're forming like the polymers of amino acids, they used a highly basic environment. And then the the dry environment for the protein synthesis was needed. However,
But you can
for the RNA
form.
synthesis, they would have things like wet dry cycles that would be needed. Whereas to keep these proteins stabilized, they need low water activity. And then
Oh,
also like
from
the protein synthesis needed like much higher temperatures than the RNA synthesis.
yes, yes. Some of
So that's
that is
incompatible.
accurate. No, it's not, because amino acids
What?
can be formed under acidic or basic conditions, right? And just because they were formed under you said basic conditions, is that
Yep,
what you said?
that's what
Yeah.
I said.
So just because the amino acid itself formed under basic condition, right? It's not going to just fall apart if it goes through from my understanding, the wet dry cycles was for protein formation.
Mhm.
So the
No,
amino acids,
RNA
the
synthesis as well.
RNA synthesis was the clays and the mineral stabilization, from
Yeah,
my
but
understanding, is how a lot of that has been done. The
that's
wet dry
stabilizing once you
cycling.
have the RNA polymers though.
No. The Montmorillonite clay actually acts as a catalyst to polymerize, to link, to perform the chemical reaction that links the nucleic acid monomers into an RNA polymer. There's a chemical that's actually contained in the clay that participates in the chemical reaction, catalyzing that polymerization.
Right. So what I'm saying is, in whatever experiment you want to reference with the creation of RNA and the creation of proteins, you'll see different conditions, different pHs and water levels needed.
Yes, because those are conditions that are found in nature, both today and hypothesized prebiotic conditions that could have been found in nature.
So I'm curious. I'm
you
curious
want to
if we're gonna
test
lay out
the
if we're gonna
reactions. Hang on,
Jace,
Dyslo.
if you will. All right.
Just let me finish real quick. You want to test the chemistry, the prebiotic chemistry, in relevant modern conditions, but most importantly, in the hypothesized prebiotic conditions.
Okay, so I
That-
think that what would be helpful is for you to like lay out a storyline of any sort, just whatever the best storyline is to favor your position, and how this would have happened.
Yeah, I'm trying to bring this to reality, right? Like having like essentially a forensic scene in nature, right? Where this event takes place, right? And we've got an arena, let's just say it's a pool, right? A stagnant pool. I don't know. What's this pool going to be consistent of? Is it obviously we got to have some ingredients.
We're in reality, bro. You can do all of this if you wanted to obtain the all the needed
outcome.
materials. Like I said, Dale, I think it's Denton. I will post it. I'll tag Taylor and I'll tag you, Dyslo. This
That's
group,
fine.
they have multiple publications that detail. They tell you exactly what to do to go from inorganic precursor compounds in relevant prebiotic hypothesized conditions to RNA strands that are encapsulated in lipid vesicles that are undergoing processes akin to biologic evolution, right? They give you the whole array of how to do it yourself.
Yeah, well, just share with me whatever that may be. Share with me the array, whatever they laid out, and then we can look at the methods of what they used.
I mean, it's one thing to question the veracity of what's said, right, within a lab frame setting, but I was just trying to create like a natural, realistic setting in the sense that this was all said, that nature's said to arbor life, right, in the creation of it. So I'm trying to frame this in my mind, right, to see how plausible and likeliness the things you say, right, with like Taylor said, the changing conditions, whether it be the pH, temperature, different ingredients, what have you. You know, these things have to be mixed in such a way and then isolated, right, in order to essentially lock in. You show me.
Nature's not
We're
doing
talking
chemistry.
about.
Nature's not doing chemistry in a lab like James Tour. You don't have to isolate. Nature doesn't have to isolate the molecules afterwards. Chemists
Okay, so then we can talk
isolate.
about that in the debate. I love that.
Chemists isolate the molecules, right? People like James Tour and sometimes myself, we isolate molecules because we can't see the molecular structure with our eyes. So we have to do something called NMR, nuclear magnetic resonance, or some other spectroscopic method to make sure that our chemical reaction, the chemical reaction that we performed, to make sure we made the right molecule, because sometimes a reaction can form multiple product molecules. So we isolate those molecules as chemists so that we can characterize them using spectroscopy. That's why chemists have to isolate them. Nature doesn't have to do spectroscopy on the molecules it makes. It doesn't have to isolate them.
So by what mechanism then does the next process take place, right? Well, with eliminating the ingredients already used and introducing the new ingredients. Like this is like the improbabilistic nature to what I'm trying to expose in this whole origin of life thing, with the fact that, you know, nature can't do all these things that you're claiming that would be required. And I'm not going to take the conversation away from Taylor, but I just wanted to throw that out there. And I'm not trying to get on your bad side, Jace, but I felt compelled.
Well,
the origin of life research seems a little shoddy because none of it's done in a prebiotic environment.
Right. Like, it's questionable even given the necessary ingredients within a lab setting, let alone
that's just silly.
taking place in nature.
almost all the experiments in origin of life research are done under prebiotic conditions, or at least hypothesized prebiotic conditions. Right. So just saying that they're not done under not done under prebiotic conditions is just patently false. And I've posted tons of research articles.
Okay, I'm not gonna like
that
I'm not going to right now. We're
show
debating
that that's
Saturday.
not the truth. Like, can we not interrupt me? All right, I've posted
Bro, you just
many
go on and on,
research
dude.
papers here. I'm sorry you don't want to hear it, bro, but I've posted many articles here that show that they do it under prebiotic conditions. So that's just a
Wait,
patently
I'm sorry. Which
false.
one? Which one did you post?
Yeah,
Let me see.
she's got a debate beast coming up with Jay, so she's not trying to spoil the goods yet, you know.
Search my name in the text chat or whatever, because I'm walking around right now, and to be able to speak on the phone, I can't search for things while I do that. So if you want to look for the
pretending like he's having the
papers,
debate now.
search my name, and then search
you've said
origin
enough.
of life. Stop interrupting. Search my name and then search origin of life, and you'll find plenty of articles that I've posted here.
Okay, so I'm just curious. Are you posting these in the live stream text?
Yeah. All right, I'll find them if you're unable to.
He has posted them in the past, Taylor. He's not posting them now. He's pretending like he doesn't want to debate, but then he's having the debate and making the statements and declarations. It's rather peculiar, Jace.
Well, I've had this debate like hundreds of times. So
That's
yes,
nice, bro,
I've
but either we wait for it or you do it now, bro.
And this isn't anything new. Like, we're not talking about anything I haven't talked with 30 other people here about. So I've posted all the relevant
we're all just a
papers
number
in
on a
the
dial for you, mate. We
past.
get it.
I don't need to
real quick,
post them
Jace,
all right now in real time. But
titles
what
of these
I did
papers?
offer, can you just let me finish? What I did
I'm
offer
trying to get you
is
your
for
paper.
her to send me some papers that she'd like to discuss in a debate, and we can do that on Saturday, like I said.
All right, cool, man. Let's let someone else speak for a little bit. Jesus Christ.
Yeah, I was gonna say
Well,
I could.
I mean, to be fair, he was being asked a lot of questions. Even though he does go on long, I mean, we still got to try to give him the respect to answer the questions. I mean, and he's taking his time to do that for us, so let's try to be cool.
That's what we're complaining about then.
No, just let other people get in and have their opinion.
But he loves to talk. We all know this, right? This is nothing new, so I don't understand why we're pretending like it's not happening.
Go ahead, Galatrax.
Anyway.
Were you gonna say something?
Yeah, get in, Gally.
Yeah, I found all the papers. I just wanted to say that, like, I can find every paper.
Cool.
What papers? I'm just gonna ask the titles of what papers he's asking to find. The RNA pyrimidine nucleosides unifying concept, or the proto-RNA as a source of biochirality. What papers? Like, which ones are we looking for?
Yeah, I'm trying to find them myself. There's like probably a good 20 or 30 of them that I posted, but I believe the ones relevant to what we were talking about before come from the main researcher. I believe his name is Dale. His first name, I forget what the last name is, but I'm trying to find those papers.
Okay, yeah, absolutely. I'm excited to talk with you about these. And let me ask you one more question. So do you study did you take like cell biology classes too, or just like focused like heavily on biochemistry? I'm sure you did, but are you familiar with I'm sure you are, right? Like with essential dogma and things of this nature?
Yeah.
Okay, good. Perfect. I'm excited to talk with you Saturday at 1 p.m. Eastern Standard, or did we say, I think I suggested that we could do 2 possibly, and then we could do it like for a couple hours. Would that work? 1 or 2? You said 1 to 4, so
Yeah.
okay, perfect. Okay.
Yeah, probably like 2-ish. Like 2
Yeah,
to
the later
4.
for me the better, because like Saturday, like usually I'm working pretty late on Friday nights, and then I'll sleep in on Saturday, so you know, whatever the later is the better. So whatever that would be for you.
Yeah. Works for me. I mean, I could pretty much do any time Saturday, but like 2 to 4 is.
Okay, we'll get it
best range to shoot for.
to start with, or are you saying you have to end at 4?
We could start at 4, but
Well, we don't have to start that late. We'll try to start at like
as
2,
it gets closer, I'll
3.
probably work out more of what my Saturday's gonna look like. So we'll just aim for between 1 and 4 Eastern time to start.
Okay, cool. Perfect. And I'll try to find those papers that you referenced, that you posted, and we could go through them.
Yeah, Discord has made it harder to search for things, of course.
Yeah, so I would just say that a lot of these, like, not all of these reactions that are used as evidence for original life research, do they acknowledge are in prebiotically plausible conditions. So, I mean, some of them actually do mention that as a limitation, and they'll still get used as, like, original life evidence, which is fine, but you just need to work out how we'd get to those conditions in order to have these, like, reactions happen if they're, like, acknowledgeably not prebiotically plausible. So that's something that
But
I
that
would look for
doesn't
in.
make any sense to me, Taylor, right? If they're publishing origin of life research in an origin of life journal, right? Their experiment is done under origin of life or prebiotic conditions, right? They wouldn't, why would they say that that's a limitation? That doesn't make any sense. That's the whole point of what you're aiming for is the prebiotic conditions. That's the most important part. Now, there
Mhm.
might be like 1 or 2, or maybe even more groups or papers that like these are just chemists in their lab. And so they don't have these like natural acids or bases. Like they just have like sulfuric acid or carbonic acid or like ammonia or something to work with. But even that, chemists know that if it works with one base that has a pKa or pKb or pH of a certain level, right? It's the chemical reaction will work with any other base of a certain as long as the pKa is comparable, right? You don't have to replicate like the exact condition. So that's the only way I could see them listing not having like exact prebiotic conditions as a limitation.
Mhm.
But there's tons of research that I know firsthand doesn't they do it under prebiotic conditions.
Right.
We
So, like,
save
for
this for the debate, Taylor? Or
what'd you say?
should we save this for the debate, or?
You probably should.
Yeah,
I was
because
just
I
gonna.
do have to take off in a
No,
minute.
no, no.
So
I got you. I was just gonna say there are a few papers, like, with the like, some of the RNA papers where they're using, like, already synthesized self-replicating polymerase, RNA polymerase, like, that they acknowledge, like, "Hey, like, this is just kinda conceptual, like, we're not actually, like, doing this on it, we're just seeing if this is conceptually possible." So, anyway, that's all I wanna say.
I feel like that's going to be the scarce minority. Like even the people that Professor Dave brings in his video, like some of the researchers in those videos, like if you just look at their work, they've done uh, certain parts of the process under prebiotic conditions. So, like having these RNA limitations, like needing a polymerase, like that must have been for a very like specific application. Like that experiment probably wasn't testing because they formed the RNA using an already existing enzyme, a polymerase, right? So they knew that that's not how it would have formed prebiotically in nature. So I highly doubt their experiment was the purpose of the experiment was to naturally form RNA. And then the researchers were just like idiots and used a polymerase. Like that
Exactly. No,
doesn't
I
make
agree
Quick
with
any
that.
question.
sense to me what you're saying.
I got a quick
I'm just
question,
saying,
Jace.
yeah, go ahead.
What do you presume? What are they assuming the temperatures to be in the prebiotic conditions?
Um I don't know the exact numbers, but there's certain data that's taken from geology they try to estimate both pressure, temperature, and chemical conditions. I don't know. You'd have to look for the specifics. I don't know them off the top of
Yeah,
my head.
well, I'm just curious because I mean, going with the model, right, that they're presuming, the Earth's crust would have taken millions of years to cool down to the proper conditions to even have life, the necessary ingredients to be being formed properly. So
Yeah,
I think,
well,
yeah,
biogenic
well, so
carbon
again,
cycles have been found already, Dice. So, like, we know that it wasn't during the Hadean, wasn't during the era where the crust was molten and chaotic. That's not when life formed. Biogenic carbon cycles, which are what paleontologists basically call the precursor to actual protocells and life and whatnot, formed after the Hadean, after the crust had cooled and all that. So you don't have to worry about that.
well, but again, I mean that's given that the timeline, right, was permissible for the Earth's crust to cool within that specifics, and that's kind of what I'm pointing out. Is I don't think they necessarily got that right, but either way, whether you agree with that or not, because that's a whole nother argument to go down to even have your ingredients to begin with. But point being, see if you could figure out what those temperatures are, the presumed temperatures, and then make sure that you double check whatever paperwork you're submitting has that accounted for within the
What are you, a
within the
referee?
experiment. Well, I'm just making sure we're holding to what's said, because if you're going to assume prebiotic conditions, right, then it's like, well, let's just make sure that we've got all the variables to those conditions being met.
Yeah, just Google like geology prebiotic conditions and just read like the top paper that comes up and see what they hypothesize or what they've measured the temperature and pressure conditions to be and the
Right,
chemical
right,
conditions.
right. And they're doing that. They're doing that within the experiment as well, right? When they're
It's not a mystery, Dice. And no, the origin of life
Bit of a mystery.
researchers don't have to go out and measure those things. They take the data from the geologist and just recreate the conditions, whether they're in the field or they're in the lab.
That's interesting because it seems like they're doing whatever would work prebiotically, like kind of like a post-diction. Like what would work? Okay, that must be the conditions rather than in any of these original life research papers, I have not seen any references to geological conditions and what like the hydrothermal vents would have consisted of and things like that. It seems like whatever's working chemically is what they're going to go with. So if you have any papers
What?
for that, I'd love to see them.
All of those things are all the composition of like black smokers and white smokers. All that stuff is known. Like just Google
let's
it.
not get racist here.
The
Yeah,
conditions,
I'd love to
some
get
of the
into stuff like that.
experiments are tested under an array of conditions because, like I
Uh-huh.
said, nucleic acid polymerization, the chemical reaction that happens that links the monomers of nucleic acids together into an RNA or a DNA polymer, can occur under a whole range of conditions, be it acidic, basic, oxidizing, reducing. Right. So these things have been tested not under just one prebiotic set of conditions or one hypothesized prebiotic set of conditions. It's there. Some of these experiments are done under multiple.
Do you have a particular experiment you're referencing there? Are you talking
Yes,
about the
some of the ones we were
format experiment
talking
with
about
Sutherland?
before. The one from that Dale guy, those guys,
You
they
keep
had
saying Dale
different
guy. I'm not
water.
sure. I haven't been able to find that one, but I would love
Yeah,
to.
I'm gonna post it in a few minutes. Like I said, I can't
Cool.
answer
Yeah, no problem.
questions and look for the thing at the same
Can I
time
have
because the
one
way
last question
Discord is on the
before
phone.
you leave? I just have one last question. Do you believe in the hydrothermal vent theory or the warm pond theory?
I tend to lean more towards that this happened in the deep ocean at first, than the warm little pond. It's more of a kind of outdated. The more modern view is that it was like I said, an array of conditions. So
Mm.
certain steps happened in the deep ocean, certain steps happened intermediate ocean, shallow ocean, some steps happen in freshwater, right? There's areas on the earth where these environments mix together, and those
Right.
boundaries are thought to be where a lot of these different steps, when you need these changes in conditions, would
Mhm.
have occurred.
Okay.
Because that's where
All
they
right, cool.
occur when we perform them in experiments.
In
That's where they
experiments,
occur.
exactly. Perfect. All right, thank you. Thank you for all your info, and I look forward to talking to you, buddy.
Yeah.
You're one of the only few people that'll talk to me about it, so I'm extremely appreciative of that. So thank you for that.
No problem. No problem.
I think JayAce is cool. I vouch for JayAce.
Well, thank
He's
you.
not. He's
Appreciate
not cool. That is retracted.
that.
I pulled up every single paper you posted, and I can't find a Dale. Do you know the name of the paper by chance? I went through 348 links.
This,
The elusive Dale.
yeah, I'll post. I'll find it. I'm gonna have to just Google it because I can't find it either. So Discord like changed how you search for things now. It's
It's
different.
probably down under.
If you
In
do,
Australia.
if you type that in
Yeah.
the search, you can look up all the links you've ever posted. I just tagged you in chat. So that'll show you all the links you've ever posted.
Okay, thanks for that.
Taylor, are you still there?
Yep.
I was gonna tell you I found that some of those footprint fossils, the reason they were cut in half or broken, because they were using it for like a compression test. Like they laser cut it to look at the compression layers back before they had a way to look through it.
Are you talking about in Glenrose,
Yeah,
Texas? Yeah. Cool. Yeah, I can't wait to go to that museum. That's gonna be dope.
I was mind blown to hear you going. That's cool.
Yeah. I'm gonna try to do like a miniature, like little documentary, trying to record everything. But I don't want to put standards on myself too high because then like it gets really stressful. So I'm just gonna keep very low standards. But I will try to like document some of these different things and compile the videos. So yeah, that should be fun.
Dude, Trey is literally this guy that I'm learning from. He just like puts a video camera up in front of his TV and he just does like a little PowerPoint slide behind him on a TV.
Yeah, I love
Yeah,
Trey Smith. I was telling Galatrax about him.
Yeah,
Trey Smith is
rocks.
awesome.
Hell yeah. What'd you say, Gail?
I said
Galatrax?
Trey Smith rocks.
Hell yeah. I just found him maybe like 2 years ago, and I'm just like, how cool would it have been to follow this guy the whole time? Like,
Oh, like from his origin.
yeah.
Yeah, I've only seen a couple of his videos, but it would have been cool to be like on that ride as he's first starting out.
Yeah, I've had countless people just like you can see him visibly like I guess disconnect cognitively because of how he looks and how he presents himself, but once you get into the content, like the way he looks, the way he presents himself is like perfect. It's hilarious. Like he's not trying to be some social media guy. He's just sharing crazy shit and traveling places.
I can't believe that he said that early on a lot of his stuff got destroyed. That broke my heart.
Same.
Man, I
Oh
can't
yeah,
imagine.
he got straight attacked. Like I've heard the story multiple times, but it seems to change a little bit. But it probably was crazy time.
Well, it might have been multiple things, but he said like he had a fire or a flood in one of his basements, like with his videos or something. That's the only one I've heard so far.
Yeah, I think it's two separate things now. I think there was a flood that he's not sure if it was, accidental or purposeful, but then the fire got to a point where he believed it was really an attack.
Aw.
Fucked up. Like you
Poor
follow guys like
Trey.
Eric. Yeah, you follow guys like Eric Dollard. Like these dudes are so
Legend.
yeah.
Absolute legend. I recently
Eric Dollard.
got recommended a playlist on the 1,000-Year Reign and the what do they call it? The Little Season. That's what I'm recommending. So I'm gonna look into that. I'm curious being about that. I don't know if
The
you guys have heard anything about that.
1,000-year reign.
Yeah. So it's like the idea that Christ has already returned and he reigned for 1,000 years, and now we're in the little season of like devil.
Is that what Trey thinks?
No. No.
Okay.
And Kingdom in Context specifically did a series, I think, anti this against. But this
There's
might be the
definitely
thing I'm streaming on AC.
nice. There's definitely something up with the larger cycles. Like if you look into is it Gematria, right? The numbers and letters. There's a second code that's integrated directly into it. So, and I've seen some common denominators. Like if you take different, like certain people say, this word should be traded out with this, or this word should actually be this. And if you use the number version labels, you'll find like every other part beginning of a verse was like amen or something. It happens to be the beginning of like 77 verses or something. Other number codes fit in with what the word should be, right? So it suddenly makes the whole code work but some don't have any tells like that. But when you start looking at it through that filter, it's kind of wild. Like it's a whole nother level of communication.
Yeah, I'd heard some interesting things about the numerology in the Bible, but I've never actually been able to find like good stuff on it. And then my question would be, is it universal across all translations? Because that would lock it in, right? Like no matter how you translate it, it would still have that. And I doubt it.
Well, that's what
I
got me
just want
looking at
to
it.
update. So the guy's name's not Dale. Sorry, I was remembering his name wrong. It's David Demers. That's his name. So I don't know, I can't find the article specifically that I posted back in the day, but if you just Google David Demers Origin of Life, right, just look through his work. And I also just Googled RNA encapsulation to find his work. There's tons of other papers here that you could look through, Taylor, if you're interested, just on that one topic. Some of them are prebiotic and some of them are just chemists or biologists in the lab messing around.
Is it called Where Did Life Begin? Testing Ideas in Prebiotic Analog Conditions?
I think that's one of his papers. Like I said, his group, they published like a series of papers going through this step-by-step experiment they did of RNA encapsulation from like prebiotic or from inorganic precursors.
I posted that one.
Again, it's not just one paper, right? This is a remarkably complex thing, right? And so they had to publish like a series of papers to detail all of it.
Right. So if you click David Deemer's name, you can get to all the papers he published, at least on PubMed. There's about 7 pages, each with 10 results on it. It's like there's 70 papers on PubMed alone. I don't know if there's another
He's
site where
done
he
a lot of.
publishes. But there's a
He,
lot.
he's a prestigious origin of life researcher, so he's going to have a lot of papers that he's done. There's also Lee Cronin. He's done some stuff with proteins, but he's also done some other stuff. He's another big name in the origin of life field, Lee Cronin.
Lee Cronin. Okay, I'm about to look him up. Yeah, like there's so many original life papers. It's hard to like read them all and memorize all the people, but you know, if there's ones that you like in particular, see, I have a problem with all of them. So like for me to send you papers is going to be kind of funny because I think that all of them have issues. However, if there's like from the ones I've seen, right? So if there's one that you particularly like or think's plausible and you can like, you know, defend that, then share it with me. Cause I don't know where I'd start in terms of sending you papers because like I said, I think they all have limitations.
Well,
so anyway.
Right? Like I get what you're saying, but the problem is the reason why I want you right the number one, the field isn't really in question, but number two, and importantly, is that it's remarkably complex. So like I want you to talk, like pick the specific topic you want to focus on, which would be done nicely with a paper that you find, right? So then we can just address that one topic rather than like an overall because even when we're just talking about these few steps leading up to a protocell, right, that is a remarkably complex string of events, right? So if I send you a paper, right, that it's going to be like that kind of thing, but it seems like you're trying to paint the idea that these papers all have problems. So I want you to find a specific paper you think has problems and then tell me those problems in the debate or whatever, and we can discuss that.
Yeah, so let me just tell you what my position is that undirected abiogenesis is untenable. That's my position. And so like I think that all of them show some sort of direction and some sort of like intervention, any of them that make significant progress. So that would be my position. So like things that can show like plausibility prebiotically that actually are relevant and can form something that would be conducive to life to move on to the next step, right? They could be used to move on towards life would be something that you should maybe post. But
you have to get more specific though, because I could just make the exact opposite claim. Like the there isn't any issues, and I can predict the things that you think are issues aren't really issues to people who are actually in those fields. It's just you're trying to simplify something that is incredibly complex, and it's in that complexity and minutia you really have to have an understanding of it to grasp the actual topic. But
right. So what I would say is that there's no like cohesive theory to how it even could have happened. So if you have a cohesive theory, like a storyline per se, as to how life could have formed, that it maybe would be our step one, because I'm saying there is no storyline. If you think there's a storyline, present your storyline and we can go over the evidence that
I
your
don't have
storyline is
to. The
plausible.
field has already done that. And I can't
Well, tell me what the field
I can't present you this remarkably complex storyline in like a few hour debate, right? Scientists have been
you should be
debating
able to.
this in origin of life research for
You should be
years
able to.
now.
Have you taken a scientific communication course?
No, there's no requirement that we boil down abiogenesis, which is, in my opinion, the most complex scientific topic that's out there currently. And boil that down
Answer,
into
you won't.
simplicity for a few hour debate. There's
So look,
no
let me
requirement to do
You
that
can answer, or you
because
won't.
the minutia in the details are where all the meat, all the good stuff is. That's where the understanding of how this all happened comes about, is when you start understanding these really deep details of it.
So
So
the
more
thing
simply
is,
put, you'd rather focus. Hang on, guys. So more simply put, JayAce, you'd rather focus on a specific part of abiogenesis rather than it as an entirety, right? That's what you're saying?
Well, I just want it to be like how we would do it in science. Send me papers that you think have issues with the data or the procedure, and we'll discuss if those are really issues or not. Let's get specific. Let's just not make these broad statements that there's an issue with every paper from that field. There's no specificity with that. I get that's what you want to be true and what you want to believe because it that supports your world belief, but we need to go through the specifics of what's wrong.
Okay.
Let's get a specific paper or two. Let's get a specific paper or two and actually go through the meat of what's wrong with the methodology or the data.
Okay, so let me just respond. So, okay, my claim, how about this? How about this claim? My claim is that there's no cohesive storyline to abiogenesis. Can you provide one as a counterpoint?
The field has already done that.
Then it should
I've
be
said
easy.
this
And that's pretty
before.
easy to do.
It's not something that you can do in a few hours debate in any great detail.
Then
right?
it's not even
All
cohesive.
we can do
So you can't do
is
it,
talk
JayAce.
about an oversimplified form in oversimplified terms, and then you're just going to make these general statements again that I
so
could
you
just
don't
make
want
the
the debate?
opposite general statement that I don't find issues, right? Or that these things aren't issues, right? So we have to go
Dude,
through the
if you can't explain it to a
specifics.
5-year-old, then it's not cohesive. It's not a good
There's
story.
no requirement in science that you explain anything to a 5-year-old or that you put it in a publicly consumable form in a few-hour debate. None of that is a requirement of science. So
Okay,
just
so
don't make these silly statements.
my claim was that there's no cohesive storyline. So if you're saying that you cannot explain it in a few hours, then it's obviously not cohesive.
What do you mean? Just
No,
because
he said the
something is
field already has done
complex
it. That's what he
doesn't
said.
mean
The
it's
field.
not cohesive.
If the field's
And
done
yeah,
it,
if you go through the research in the field from people who do the systems chemistry and the autocatalysis research, to the basic chemists who are doing the inorganic chemistry and the organic chemistry, right? If you can start there and you can go through that research all the way through all the steps up to protocells and through how those start to go into biological evolutionary processes and how actual structures, biological structures came about, like the Golgi body, Golgi apparatus, the endoplasmic reticulum, the smooth and rough, those things, right? You can go and read the research about all that stuff. The field has already painted that picture, and it's very complex and requires an understanding of a lot of things to really understand. And I'm not going to be able to do it justice in just a few hours debate on Discord, and there's no requirement for me to either.
is there a cohesive storyline or not?
I thought the story was on.
Do I have to make it any? All right, yes is the answer, and I've
Yes,
answered
there's
that
a
many
cohesive storyline.
times now.
Then you should
Can
be
you
able to
explain
I
summarize
do
it.
have to take off though, so I guess I'll see you
He
guys
can. He doesn't
later
want to.
in the
That's
week.
that's what he's saying.
Thanks for taking up all the mic time,
Just go
Jace.
read the research, man.
I
Scram,
thought
buddy.
the whole story was given. I thought we can watch
Yeah.
the Big Bang Theory, just the opening. Isn't that the story? You couldn't have done that in all those words you said.
He's talking out both ends, man. Really. Completely
I could sing the
logical, but
song maybe.
inexplicable, apparently.
Okay, well, I guess we'll talk on Saturday.
I don't know. I mean, he usually can. I mean, I thought he could usually give the whole like the story. Like you got the single cell organism, then something happens, and nucleotides, and something. Like, I don't know why you couldn't do it.
There's a difference between can't and won't. He just doesn't want to do it. But he said the
Yeah.
field has already done that. So I guess you can just. I guess you could just get a summary from the fields, Taylor, since he doesn't want to provide one for you. That's
right.
Weird.
There's
It's like about
scientific communication courses that we should take, that we should be able to explain scientific consensus and knowledge to individuals in the public. So if you're like refusing to be able to do that, then how much are you like? Then how good are you at understanding the science if you can't be able to break it down and explain it? Like it's just you
I
should
mean,
be able to explain things to the public.
and Jace, you got to
like
remember,
if I
she took this over to Gem Pandas and whooped the whole room. So you got to get yourself together, get it together. He left.
no, that's
I wasn't
fine.
there
Yeah,
for it.
but does the statement though that the field is already like what is he saying? The field has already determined all of this or something, which would imply that it's a simple. I mean, if you said that about any other field, you just say, okay, I can go cut and paste the information, bullet point it out, and here's your description. But at the same time, you're saying that the field,
ChatGPT, bro, right?
yeah. But like then at the same time, you're saying that it's like he didn't quite say that it wasn't possible. I think he was sort of inferring that it would take too many hours or it'd be such a laborious point that it's kind of, is it worth the time or value, I suppose, was his inference.
Which field specifically? Didn't he touch upon the fact that this might be multiple fields, like chemistry, biology, a whole bunch of different fields? So which field specifically was he referring to?
I'm not sure.
I don't know.
The origin of life in its entirety.
We'll just tap into the field, see what they have to say. The field of science.
Yeah, well, I mean, I don't know, if I'm being objective and I step back from it, like if Taylor had a more poignant question, then maybe it could have been addressed. Just to say it's not like your whole thing sucks, it's not cohesive. And it's like, wow, what do you want me to do? Start at the beginning and go to the end? It does take a long time.
No, it shouldn't. You should at least have like some environment that it happened in. Like, oh, it happened in a hydrothermal vent, and then or it happened in a in a warm soup or whatever. I wasn't asking for the details.
Yeah, I mean, I think it would just be it happened because we have life, so it happened somewhere.
Yep. Was it directed or undirected? How did it happen? These are just basic general questions.
It was undirected. I'm just playing along. I don't really have these answers, but undirected.
Or then you need to demonstrate that, like how that's plausible.
Well, it's plausible because the universe is all things, and all possibilities rely on all things. And once they, were separated, they can come back together. So it's always possible.
Right. So then,
no thanks.
Those are statements, but you need evidence. That's about it.
I'm saying, if you got the universe, you got all things, and you break it apart, and you got a little piece over here, a little piece over here. All that had to happen was like a little lightning strike or something.
Huh. That's weird. That should be easy to demonstrate then. So, like, if we continue trying to jump higher and higher, will we eventually be able to jump to the moon with enough time?
Awesome non sequitur.
Yes.
Well,
if we're
this is
standing
the
on
thing.
Merlin
Like
Point.
with the universe and all the things there, right? It's bound to happen. But there are certain things that are like that you can't overcome. That are basic universal laws and principles.
Yeah, the possibility that I referenced. I'm saying that the possibility, right? Like it's either definitionally possible or impossible. Obviously, it's possible because it happened.
But it's possible. Is it possible undirected in prebiotic conditions?
I don't know. I don't know enough about this stuff. Somebody else is gonna. I'm just playing along.
Well, if it's a primordial soup that has a hydrothermal vent that happened in panspermia and RNA somehow manifest through the Merlin clay, then yeah, simple.
Panspermia of what?
I'm just being facetious. I just tapped into Chat GPT. Where did life come from? And there's 5 different hypotheses, and then it ends with still a mystery. Despite these theories, scientists still are trying to answer how non-living chemicals turned into self-replicating, evolving organisms.
Taylor's excited. She's like, I can't wait till Saturday. Are you kidding me?
What's going on Saturday?
Is it a formal? I guess I don't know if it's a formal debate, but just a debate between JayAce and Taylor.
All right. Yeah. Like just agreed upon now, because if it was just agreed upon now, it probably won't happen. Just knowing Jace.
Can you guys imagine a universe without life? Like it's just this sort of inanimate matter flying all over the place without anything to observe it or